My name is Christoph Gerber. We are here at the Swiss Nano Institute, and we started that institute in 2001. It covers topics from quantum information all the way to life sciences. So ever since we developed the atomic force microscope, I stayed in the field, and it started actually the field of nanomechanics. And in recent years, we have taken this technology way beyond imaging and developed a diagnostic tool with a wide range of applications in personal healthcare. So what we're going to do is we're going to show you, we're going to take you to the labs and going to show you how this technology works.
My name is François and I'm a biochemist. We develop biological sensors for various biomedical applications. Among them is the analysis of cancer. For this we use special micro cantilever arrays, as you can see here on the screen, with 8 cantilevers. They start from here, from this groove, and have a length of 500 micrometers. They are 100 micrometers wide and from 300 nanometers to 4 micrometers thick. For the purpose of the sensor, the cantilevers are gold-coated, which allows the coating with special biomolecules for the detection of various cancer markers. The molecules we are using for the detection are DNA and protein molecules. The functionalization happens in a special device. I'm now mounting the cantilever array onto the functionalization stage. The functionalization stage contains 8 microcapillaries to ensure the coating of the cantilevers with biomolecules. Each capillary is equipped with a microreservoir that contains the liquids. The microcantilever array is now placed into the measurement chamber, which is mounted into the optical system, which contains a laser unit and a position-sensitive detector that monitors the bending signal of the cantilevers. The measurement chamber is also connected to a vessel containing the sample which we want to analyze. The whole system is placed into a temperature-controlled box to ensure a stable temperature during the measurement. I can here now start the measurement. I show you the analysis of different experiments with samples from skin cancer cells and non-cancer cells. As you can see here on the graph, depending on the time, the development of the bending signal after injection of different samples. Here we are looking at the differential signal between the probe cantilevers and the reference cantilevers. The black curve shows you the bending signal after injection of cancer cell samples, and the green curve shows you the signal after injection of non-cancer cell samples. The blue and the red curve in between are different ratios of cancer cells sample and non-cancer cell samples. The difference between the cancer cell samples and the non-cancer cell samples is a single point mutation in the gene responsible for cancer development.
My name is Hans-Peter and I'm a physicist. You've seen before that we measure with cantilever arrays. This is a well-established technique, but here I want to show you a new method of more compact sensors that are piezoresistive membrane sensors. A piezoresistive membrane sensor consists of a membrane that is held by 4 bridges. These are piezoresistive bridges, and if the membrane bends, we will have a resistivity change at these bridges. This format is much more compact, and we can measure more accurately than with the cantilevers. So This is fabricated at EPFL in Neuchâtel and looks like this. Here you see the membrane made of silicon, and here the 4 piezoresistive bridges. We not only have one of these membranes, we need a whole array of membranes to do the measurements. In this case, we are also investigating a medical topic. That means we investigate the breath of patients that are suffering from a disease, and from the pattern, of the response pattern of the sensors, we can deduce some results about the disease. But first of all, we have to coat these sensors with a polymer layer, and we will do that in the lab next door. Here I want to show you how we functionalize these piezoresistive membrane sensors using the inkjet technology. We eject from a nozzle droplets of polymer solution onto the sensors. First, I want to show you in a stroboscopic view how these droplets are ejected. Here you see the droplet being ejected from this nozzle, and I can change the delay time to move it upwards or downwards so that you can actually see how these droplets are ejected from the nozzle. In the next step, I really want to show you how we coat the sensors. For that purpose, we go to a different camera view. Here you see these sensors, these membrane sensors on the chip and the nozzle right above it. When I start now the ejection process, you see how this polymer solution is being deposited on these membrane sensors. Here the nozzle moves down and ejects the droplets onto these membranes. One after the other is coated with polymer. So now we're back from the lab. I have mounted the sensor already into this little box, and I will show you how the measurement will take place now. So for these membrane sensors, for breath testing, we use such breath sampling bags, and I will fill these now with my own breath by just taking here this mouthpiece and then blowing into this bag. Okay, so this is about 1 liter of breath in this bag, and you see it is a completely non-invasive method. So this will not harm a patient, it will not take blood or anything else. So it is really a non-invasive method to investigate diseases using the breath of a patient. So this bag is now being connected to this little tube here with the injection needle, and then the breath is being transported by a micro pump into the sensor chamber, which is inside the box. So I put this a little bit aside, and now you already see that the signals are changing here on the screen. We will do now a couple of injection purging cycles to record several samples of this breath, and then it will be investigated using an analysis method, as I will show you right away. So here I show you the results. We've done 10 injection cycles and purging steps of different conditions. So one is just my own breath, one is after drinking some wine, and we have also 2 reference calibration samples, ethanol and water vapor. So you see these 10 injections and purging cycles. For analysis, we do a mathematical transformation, a so-called principal component analysis, which gives you this two-dimensional plot with several clusters. So here, this is the cluster showing the measurements of my breath here after drinking some wine, and here the control samples, ethanol and water vapor. Now we have shown you a few of the possibilities in the wide range of application that are offered by this method. Finally, I want to address the attention to this commercial setup that is operated by Jiayun Zhang. This has been built by a spin-off company of the University of Basel and is a nice demonstrator of how the technology can really be converted into a product. So, thank you for listening and goodbye.